From ARUP Laboratories on the campus of the University of Utah, welcome to the LabMind podcast. I'm your host, Dr. Brian Jackson.
Today is Monday, the 21st of July, 2025. We are recording this episode in the middle of the summer, which means it's the middle of summer blockbuster movie season. And there's a whole slate of films out there about existential threats to humanity, such as bioengineered dinosaurs, rogue AI, and last but not least, deadly viruses. So what better time for a conversation on real-world threats to humanity, specifically emerging infectious diseases? And we have an excellent guest for this. We have today Dr. Benjamin Bradley. He is Medical Director of High Consequence Pathogen Response, Virology, and Molecular Infectious Disease at ARUP Laboratories. He's Assistant Professor of Pathology at the University of Utah School of Medicine. Dr. Bradley received his medical degree and PhD from Tulane University School of Medicine, followed by pathology residency at the University of Washington, and then a clinical microbiology fellowship here at the University of Utah and ARUP Laboratories. Dr. Bradley has received a number of early career awards for his research. For example, the Jacob Ambrose Stork Prize, the Strandard Clayton Award, and the Paul Strandard Young Investigator Award. I also want to mention that Dr. Bradley will be presenting a free webinar on September 25th entitled Emerging Viral Diseases, a 2025 Update for Laboratories. So check out the ARUP website for more information on that. Ben Bradley, welcome to LabMind.
Thanks, Brian. Great to be here.
So there have been a lot of emerging infectious diseases in the news over the past, couple of years, let's say. Is there anyone that's been particularly on your mind lately?
Yeah, so I think that's part of the job is to keep these things in my mind. And maybe before we get into this too, I just want to take a step back for the audience and talk about what an emerging infectious disease is.
As opposed to garden variety infectious diseases.
As opposed to what we deal with every day. So with emerging infectious diseases, we're really talking about pathogens that aren't commonly spread or commonly seen within human populations. So there may be occasional spillover events that we'll discuss, or there may be a novel virus that emerges. But in addition to emerging viruses, we also have this phrase known as reemerging viruses. And that typically speaks to things that maybe were once a very common human pathogen, like measles virus, that we haven't seen for a long time, that are now starting to reoccur. So there's emerging and reemerging infectious diseases that we worry about. And for me, as a laboratorian, as a clinician, really my focus is on those pathogens where we may need to scale up our testing or improve our testing or develop a new test entirely. Things that I've kind of been worried about lately for the last couple of years. We had Mpox a few years ago. It's still going on. We still get positive cases for that. Also, H5N1, also known as bird flu, and then measles virus as well. So a little bit of the new and a little bit of the old.
Why don't we start with the avian flu, the bird flu? I have to confess, most of my podcast feed coverage of this has been related to high egg prices, which is probably not the issue that's driving you the most, but something that keeps it in the general consciousness. What are the main diagnostic issues that you deal with bird flu?
It's a influenza virus, so it's related to the influenzas that we get seasonally. H1N1 and H3N2 are the seasonal ones, but we've seen emergence of this novel avian influenza virus that's spilled over from wild birds into both poultry and cattle, dairy cows as well, that have then caused to infect humans. So when we think about kind of the testing that's available for that virus, it's actually interesting because some of the pre-existing assays that we have available for influenza A virus, so let's say you feel like you have the flu, you go get tested at a lab, those assays actually target a portion of the M gene that is highly conserved across all influenza viruses. So we can technically detect bird flu with those existing assays. Really, the challenge we have here with bird flu is doing that subtyping identification. So to be able to say, okay, I have an influenza A virus here. Is this one of our seasonal types, or is this novel H5N1 bird flu?
So my understanding is there have been very few human cases. Is that correct?
Correct. So The overwhelming majority that we've seen in the U.S. have all been associated with human exposure to either sick poultry flocks or sick dairy cattle. So these are individuals who were exposed to an animal that probably had a very high burden infectious dose and were then exposed either through aerosolized particles. For some of the dairy workers, they thought splashes with the infected milk. Bird flu infecting cattle was really interesting just because we've never really seen influenza viruses in cows before. And when these cows got sick with bird flu, what we noticed is they had a very high concentration of the virus in their milk. And so individuals working with these animals were believed to be exposed through that way. But you're correct, we haven't seen human-to-human spread of this H5N1 strain, which is really reassuring from a laboratory standpoint, just because that's one of the biggest red flags that I could think of where if I didn't have a test already, I'd be running to try to develop something, get something ready, because that really is kind of a signal that this is something that our clinical community is going to need access to.
So who actually does the agricultural testing? Does CDC do that, or does some other federal agency, or are there other labs that?
Yeah, that is primarily overseen by USDA. USDA has their own laboratory testing services. There's a lot of politics involved with this too, just like there is with human testing, about the cattle lobby not wanting their herds to be known to be positive for H5N1 and having some of their resources quarantined from that. So yes, there's an entire other microcosm of testing that exists out there for animals.
So basically, it sounds like you've got a situation where a few humans, mainly people who work on these farms, have been infected. So there is a need to be able to identify, is this a human infection? But there are very few of them. Does that create challenges for the laboratory in having a test ready that you may never use or you may run one of in the next year?
Yeah, I think it's definitely a conundrum, and it's something that we realized was maybe a shortcoming with SARS-CoV-2. I mean, everything, we're still not so far away from that we can't reference everything back to it. But I do believe there were some lessons that were learned both at the level of ARUP, but also nationally. And I think one of the things I'd like to mention here is that the CDC has established a contract with ARUP laboratories and a few other labs to provide what they're calling warm-based testing capacity, which basically means that we hold on hand a little bit of our lab testing bandwidth for a potential emerging or re-emerging pathogen. And also within this contract, there's an option for them to reach out to ARUP or other contract holders and say, we need you to develop a test for this pathogen. Can you do that? And so for H5, the way we were able to develop that was in part with some of this assistance from the CDC to bring that on early. And so I think those approaches of reducing the risk to the laboratory for developing this by providing some kind of financial support is very important moving forward to address that, to say, here, we're going to give you a little bit of funds so you can develop this so you're not having to eat the cost yourself.
So any particular challenges that you ran into in the course of creating the H5N1 test?
Yeah, with developing any kind of assay for an emerging pathogen, I think there's a few sort of high-level challenges that you face. And one is that there may not be any pre-existing assay out there in the literature to find. And so you're going to have to go and potentially look at deposited sequences of this virus, do the alignments, build the assay yourself. We've been very fortunate in the post-COVID world that there's a lot more sequencing capacity to sometimes see these sequences, but it may be you're using a manuscript that's 10 or 15 years old that was an academic paper about how to detect this pathogen that you're having to bring on now. There's the more practical concerns, too, of doing a validation. When you're doing this for an infectious disease, really you want to have as close to the source material as possible. And what I mean by that is having a positive patient sample or having that virus within the sample for you to test. But in a situation like bird flu, when we were starting to develop that test, maybe there was 20 cases in the U.S. to begin with. So getting our hands on that material was challenging. Add to that some of the regulatory barriers here. H5N1 is considered a select agent by USDA.
Let's define what select agent is.
Yeah. Essentially, this is a pathogen that could potentially have bioterrorism implications or is highly pathogenic. And because of its perceived threat to humanity, there are certain documentation that you have to have and a license that you have to apply for and hold. And that comes with it a lot of paperwork, a long time to get approved. And when we're thinking about trying to respond quickly to a pandemic, having to go through that entire process would be extremely challenging. Fortunately, something that USDA did in response, they put a scale back to the select agent rule for bird flu for a period of 3 years so that labs could handle it. But this virus is still a biosafety level 3 pathogen. So if you wanted to grow it up in a lab, you need a lot of special equipment. We really kind of had to work with some of our colleagues in academia and in government to get inactivated virus to the lab that we could test. So having that kind of material was another big challenge for us. And I think the last challenge that we faced with this assay was the question of when do we want to go live with this test? Certainly, we don't want to put anything out too early because then the idea is we're going to be testing a lot of the worried, well, if there's only 20 cases for this and I start testing 100 samples a day, what's the odds that a detected result is going to be some false positive just because of the high pretest probability of a negative? So we had to be very careful in thinking about that too. And for us, the trigger we decided for bird flu was when California issued a state of emergency for bird flu, as well as when the CDC put out new guidance suggesting that hospitalized individuals with influenza A should have subtyping performed on their samples.
So let me ask you a couple of fairly naive questions. Why can't you just send these patient samples to one of these agricultural labs that has been running testing on cows and chickens? And a related question to that is, if it's important to run the test in a human clinical laboratory, why can't you just validate using specimens from infected cows and chickens?
That's a great question. As a clinical lab, we are doing patient testing and we're held to a certain quality standard through our accreditation practices. And that testing needs to be done in certain facilities that meet those accreditation guidelines versus sending it to USDA or a lab that's maybe not accredited by CLIA. We run the risk of not being able to report those results in the patient chart, that there being a lot of questions about the accuracy of this testing along the way. By performing the testing in-house, we have a higher confidence of our results and feel comfortable sharing that with the patients. As far as the material goes, that really gets into a little bit more of a gray area with validation for emerging infectious diseases. And a lot of times we really are in a situation where we kind of have to make do with what's available to us. And so there are situations where we may be looking for virus that came from a cow to use to help validate our test, just knowing that that virus is still present.
So it would be the same virus, but I could imagine that the pre-analytic portions of this, the specimen collection, are going to be very different. The concentrations might be quite different.
Yeah, I think there's a lot of analytical challenges that we have with that. And certainly when we're doing these kind of assays, we really want to find as close to a positive human specimen as possible. And when we're not able to achieve that, typically what we'll do is use the best material available to us, and then we'll perform what's known as bridging studies, where we'll get that new, more accurate material, more representative of a patient clinical sample, and repeat some of the accuracy studies, repeat some of the reproducibility studies. And I'd say that's pretty much in keeping with the messaging that we've seen following COVID as well, where there really was a message of use the best material that's available to you, but also understand that as this pandemic evolves, as more resources become available, we may ask for more stringency in the validation materials you provide.
So at this point, is your laboratory receiving samples for this test, or it's basically on hold pending public health emergency that might come up?
We are still running this test. It's fairly low volume right now just because, as you're aware, it's not really respiratory virus season right now. But we are certainly anticipating when flu season rolls back around, there will be some renewed guidance from the CDC suggesting whether we need to be screening for this virus or not. And to have that assay available, I think, will be important, again, as a reference lab, we're kind of in a very beneficial position for doing some of this low volume testing that smaller labs may balk at just because they couldn't support that kind of volume.
So I guess the worrisome scenario here, like you mentioned, would be if there was some genetic change to the virus that would cause it to be human transmitted. So if that happened, then what you're saying is that we would have testing available right from the outset?
Yes, exactly. We would have this test available. It's something we'd be able to deploy rapidly. And when we were designing this test, we tried to be proactive in thinking about what characteristics of the virus might we need to account for. And just to provide you an example, let's compare something like Mpox to something like influenza virus. So Mpox, that's something that's typically transmitted by very close skin-to-skin contact, humans really coming in contact with one another, versus something like influenza virus that spreads through the respiratory route. So we know that that virus is probably going to transmit more easily into a higher number of people.
A lot more easily, right?
A lot more easily. And so because of that characteristic of the virus, when we think about the testing capacity we have with something like bird flu, we want to be able to scale that very rapidly and account for a large number of tests. And when we were validating and developing this assay, we developed it on a automated instrument platform that's able to do nucleic acid extraction, amplification, and reporting kind of all on board the instrument. And that's in contrast to the classical way of doing molecular infectious disease, which is you extract on one instrument, you amplify it on another, and then you have somebody sit and look at all those amplification curves and manually report them out. So the advantage of these automated instruments is obviously shorter turnaround time and much higher throughput. So we would be able to support that kind of pandemic type surge that we would see with a respiratory virus.
So let's pivot now to another disease that has been even more in the news lately, namely measles. So you mentioned it's not that measles is new per se, but we're definitely seeing a lot more of it this year than I think either you or I have seen in our lifetime. What are the main diagnostic issues with measles?
If you look in a textbook, they're going to tell you that measles is mostly a clinical diagnosis. And that's because typically this virus that starts off as a respiratory infection, it then disseminates, presents with a very characteristic rash. It's called a morbilliform rash. It's something that starts at the head, kind of moves down to the extremities and resolves in that direction too. These kids often start with a fever a few days beforehand. But the challenge with making a clinical diagnosis is while you may be very accurate in saying that this child or this person has measles, what makes it difficult from a public health perspective is that by the time they develop that rash, they've probably been infectious and able to spread the virus for 4 days. So even though you're able to diagnose it accurately from a public health response, from an infection control perspective.
And to treat the patient.
And to help manage the patient, you're right. So there are no specific antivirals that we can use for measles virus infection, but we can certainly give some supportive care to those patients. So for those reasons, we really want to have laboratory testing available to help with that. And the traditional way of doing this has been through serology, mostly IgM antibodies. These are antibodies that are typically formed very early in infection. But the challenge of IgM is that it's not the most sensitive or the most specific at times. So with that IgM antibody, it can sometimes take, again, up to 3 days after the onset of rash to have detectable antibody levels.
It's even slower than diagnosing by rash.
Right. So it could be slower than diagnosing a rash. And on top of that, these IgM antibodies sometimes can persist for a very long time, or in the event that a patient has a different viral infection, say they have something like EBV infection, sometimes that just ramps up the body's antibody response globally, and you may detect these IgM antibodies as just a false positive signal. What we really want to be able to do to detect measles virus accurately in this current day and age is molecular testing, PCR, NAT testing, nucleic acid amplification testing. And molecular testing, for the most part, has really been limited to public health labs, CDC, few state public health labs, and a few reference labs as well, where that testing could be done.
But again, understandably, because the volume of measles has been quite low for the past 50 years.
Exactly. There hasn't been as much of a need. And again, just to update our listeners on the situation with measles right now, we are at an all-time high of measles cases since it was declared eradicated in 2000. There are multiple outbreaks that are going on across the country. The one in West Texas is the largest cluster, but there have been many more cases where they become imported through somebody flying into the U.S. that then gets exposed to a population of under-vaccinated individuals. And that's where the molecular testing can really come into play for timely identification. We do have to be careful about some of the nuances of measles virus testing. A lot of the tests that are out there right now just detect measles virus globally. It tells you if measles virus is there or not. But what makes the diagnostics more challenging in an outbreak setting is that individuals may want to be vaccinated in response to this outbreak. People kind of are starting to get nervous, realizing that it's something significant. And the vaccine that we use, the MMR vaccine, actually contains live measles virus. It's this genotype A measles virus. So what can happen is an individual who's been vaccinated can also test positive for measles virus if you're using this generic primer probe set. And so there's been a second primer probe set developed that's specific for the vaccine strain specifically. And the test that we're running at ARUP actually uses both of these targets in multiplex to not just determine, does this person have measles virus, yes or no, but is it likely to be the vaccine strain or wild type? Because people who test positive for the vaccine strain aren't considered infectious. They're not considered a threat to spread to other people. They're not at risk for developing severe measles disease.
So one more question on measles, just going back to the way you introduced the topic a few minutes ago. I do remember the medical school lectures in the 90s and the photos of the different specific type of rashes. At least I remember having had the lectures, but there's no way that and I'm not a pediatrician, but even if I were a pediatrician, I doubt that I would be competent in diagnosing a measles rash the way that a 1950s pediatrician who saw these every year would be.
And I think it's a diagnostic challenge for our physicians nowadays who just have this ever-expanding body of information to keep up with that, we really haven't seen measles in the community since the 1950s, and I don't think there's any practicing physician from the 1950s still out there. So when doctors see a child who has measles, unless they know that this kid's been somewhere where measles is circulating, they're probably going to think about a different childhood rash, adenovirus, roseola, something like that. So again, there's a risk of misdiagnosis here if we're just using clinical approaches and not relying on our lab tools as well.
I could also imagine a diagnostic error in the other direction. So for example, a kid in Texas who gets roseola today, I wonder how many of those kids would get inappropriately diagnosed as having measles if you didn't have a specific test available.
Yeah, exactly. So really having this assay in this outbreak phase where we can also help separate out wild type from vaccine, at least the feedback we've received is that this has been very helpful to our clinicians along the way.
Let's talk for a minute about the different kinds of laboratories in this big laboratory ecosystem that exists out there. Your lab is a reference laboratory which serves hospitals around the country. So that's one type of clinical laboratory. There's also the hospital laboratories that are out there, which serve their local populations, but are less likely to have highly esoteric tests available on site. There are public health labs like CDC, which you mentioned. Well, let's just start with those. So how does a lab director think about the calculus of spinning up a new assay? If you're in a hospital lab out there, are you going to be working on measles today, for example?
Yeah, I think that's a really challenging thing. And I think we're still trying to figure out a lot of this. And there's multiple relationship dynamics that are going on that make every lab situation unique. Certainly for the community hospital lab where they need to do their best just to make sure they're getting troponins out on time, to ask them to bring on these kind of bespoke systems, I think is a little bit too much. And that's what our public health infrastructure, what reference laboratories should help support. I think different state public health laboratories, different city public health laboratories all have different capacities, expertise, and focus along the way that will help them bring on certain tests and maybe hold off on developing other tests. So, for example, in the state of Utah right now, if you were looking to get measles virus testing done through the state public health lab, they would need to send that specimen to California for the testing to be done because we don't have that capacity right now. Whereas some other labs may have that capacity to bring on. And then again, we have this backdrop of the CDC, who also is able to bring on very esoteric, bespoke assays to help out. There has to be this constant conversation. And ideally, I think it's led in sort of a national level, top down, to say, what is the capacity that we have right now for this pathogen? What is our anticipated need in the future? And what events would trigger us to expand this testing out more over time? We've talked about some of the ARUP assays we have that are very automated. If we look at how these are performed in some other labs, including some public health labs, they may only be able to run 19 or 20 samples per 96-well plate. So kind of a low throughput, but still some capacity to do the testing. And so figuring out how does that testing environment evolve over time in response to an outbreak or the threat of an outbreak is something that we're still working on and learning.
Now, public health labs historically have filled a lot of these needs, right? For obscure agents. And I'm maybe not thinking so much today about epidemic agents, but maybe low volume things like, maybe hantavirus or, tularemia might have a few cases. I don't know how many cases there are in the US each year. But those kinds of things historically have been done at public health labs, right?
Yeah, exactly. And I think kind of understanding the environment of how actionable are these clinical results to patient care. For example, at ARUP, we can do a lot of phenotypic testing to assess the antimicrobial resistance of a bacteria against certain drugs, but we may not be able to do whole genome sequencing of that bacteria to tell you it's resistant to this drug because it has this exact gene in it. And the argument would be that knowing that doesn't really affect the patient management, their prognosis, their diagnosis, because we know that it's resistant. We just don't know how. And with public health being able to do that testing, it still generates important information. It lets us know maybe we need to be concerned about some new transmissible element between bacteria. And it's also of a service to the patient because by doing that testing at public health labs, we're not in a situation where we're doing testing that we need to bill for a patient. There were some challenges for that even with H5 testing, because with bird flu, we know that it responds to the same drugs that seasonal influenza does. And so doing this testing also has a little bit of a epidemiological bend to it for the patient.
Now, at the other end of the spectrum, another category of laboratories that we haven't really talked about today are academic research laboratories. I'm guessing there are a lot of virology laboratories out there that study some of these agents. And maybe let's tie this back to COVID. Hopefully we've learned some lessons from COVID. But early in COVID, were you actually in Seattle at the time?
Yeah.
So if I remember right, there was some of the earliest COVID testing in the US was actually in a research lab at probably your university. But then there were issues with that not being a CLIA lab, and so CDC shut that down, or what was the story on that?
Yeah, I don't think I'm the best authority on this story. My understanding of it was a flu surveillance group that was prospectively collecting samples from patients who had respiratory symptoms. And then when COVID began emerging, they said, hey, we've got these samples where we can start looking for this virus early on to develop those tools. But you're right. I think there were some regulatory challenges, but fortunately I escaped too much involvement in that.
Yeah, I can't remember if it was FDA or CDC. I guess the lesson there is just because you can do it in a research lab doesn't mean that it's ready for clinical prime time.
Yes, exactly. Certainly early during COVID, I think lots of folks were surprised to figure out the number of regulations and accrediting bodies that we have to deal with in clinical laboratory medicine and sort of our quality control programs, the stringency of our validations along the way. So there is sort of that level that we need to have in our clinical testing in terms of controls, documentation, standardized procedures. But I do think those research labs can be a nice starting off point for certainly a lab to bring on an assay, again, to say, I need to start validating an assay. Is there anything that's already existing in the literature that's been done in the basic sciences? I think the other thing that these research labs can be of huge support for are going back to getting the material you need to do a validation study. So looking at our measles virus assay, we don't have a lot of measles virus on hand at ARUP, but fortunately I have a colleague who I'll shout out, Ryan Relich at Indiana University, who has a virology lab, and he was able to culture 8 different genotypes of measles virus for us for purposes of validating our assay so that we can make sure that it's behaving appropriately for that vaccine strain that it needs to call out, as well as 7 other genotypes, including the genotype responsible for the current outbreak. So we really felt good bringing that assay on because we had seen how robust it was to those different genotypes, and having those kind of partners that have that specialty lab available to them that we don't often have in the clinical testing lab, where our job is to really kind of standardize things, do things at high volume, do it quickly, do it rigorously, was a huge benefit to us.
So I'm hearing a story of collaboration across the academic side, the reference lab side, the public health side, creates more capability and capacity than any one of those sectors could do themselves.
Mm-hmm. Exactly. Yeah.
Okay, one more category of testing I wanted to ask you about. About a week ago, I saw a news story locally about measles virus being detected in wastewater in a community a little bit to the south of here. So is that testing that clinical laboratories ever do, or how does this testing get done?
Yeah, so that testing is, again, kind of part of what I think makes public health such an important partner for the clinical labs. In infectious disease, we have this idea of kind of a One Health approach. And essentially what that means is a way of thinking about infectious diseases that says we don't just need to account for the pathogen and the person it infects, but factors like the community, factors like the environment, how things like climate change, movements of people, movements of animals can sort of affect the ebb and flow of our exposure to a pathogen. And so having those public health partners who can do things like wastewater surveillance to understand what's going on in the environment, where might there be cases, helps inform the testing that we do as well. Because if we're seeing a uptick in a virus detection in wastewater, especially in certain communities, that can help focus either public health or hospital awareness to those populations. It may lead a provider to say, hey, this kid with a rash that I thought was roseola, let me do some measles testing for them as well. So having that kind of context too, I think, is very essential, and also sometimes influences what happens in our lab. So I think 2 or 3 years ago, there was a detection of poliovirus in the wastewater in New York. And in response to that, what we saw in our lab was a huge uptick in the volume of people who wanted to get tested for their poliovirus antibodies to make sure they were vaccinated. And so these kind of environmental studies that happen, even if it's not saying we have an outbreak of polio right now, we still feel those effects in the lab when they happen.
If there were an outbreak of polio, I would certainly want to see a huge uptick in testing.
Yes.
So any listeners who are within clinical laboratories are deeply familiar with the whole LDTs, laboratory developed tests topic, and how that's played out over the past few years. Talk to me a little bit about laboratory developed tests for emerging infectious diseases. Is it important to be able to develop LDTs, for example?
Yeah, so in the clinical micro lab, there are a few big groups, our transplant-associated viruses, EBV, CMV, respiratory viruses, where there are FDA cleared or approved assays. But for the vast majority of things, especially these esoteric pathogens, these are all run as LDTs or laboratory developed tests, where we're needing to source some of the reagents ourselves, understand the performance characteristics ourselves, and do a very robust analysis of the performance of these analytes. And I think it's pretty safe to say, without sounding too jaded, that there probably will not be commercial manufacturer solutions to these testing needs. And I think that's just a simple principle of market economics, is that there's often not a very high demand for these. They're run in sometimes low batches. And so the requirements of a manufacturer having to submit to the FDA, having to maintain all of this kind of QC is just too burdensome for them to really move through. And we actually saw a little bit of that with Mpox, where there were some manufacturers who initially put out some kits, but then never really moved it through the full FDA approval pathway. And so those tests never really came online. I think that's just the reality of infectious disease testing, is that I think we're probably one of the largest beneficiaries of LDTs, and that we can make what we need when we need it and do that quickly.
I also want to point out something that you said a few minutes ago when we were talking about influenza, that just because it's an LDT doesn't mean that you're not using commercially produced devices.
Yeah, exactly. So especially with some of these open access instruments now, we are using some of that same technology that FDA cleared assays are run on. And again, some of these reagents are produced in the same labs that make FDA cleared reagents. So we do have a lot of, I think, quality control checks on these products. This is not something that, we're doing in the back of the lab with gloves off and those kind of things. I mean, we have great process controls in here for these kinds of tests.
So imagine another COVID kind of a situation where a completely unknown virus comes out and starts causing disease. How fast these days can you spin up a new molecular assay for something like that?
So pretty rapidly. I mean, we have to account for a few variables here. Like I mentioned, if there are already designs that are present in the literature, that can be a huge acceleration to our ability to already order prefab reagents to run on our testing platforms. But for something that is much more novel, we will need to kind of go into those genetic sequences that are available. And even very early during COVID, we were able to see some of those sequences to start thinking about how we would design a test. There are lots of, again, research bodies, the CDC, the WHO, that put forward their own designs too, that we could double check. And so that part of it is one part of this go live. But for us as a reference lab at ARUP, the other parallel event that we have going on is kind of our what we call our clinical IT build. So building out the test so that it's present in the EMRs of our clients, of our hospital system, so that this can be ordered and reported, so that we're not faxing people results or having them fax us accession forms along the way, which was kind of some of the early struggle for COVID with some groups, was just not having that interfacing built out. So in the event, whenever the next pathogen comes forward that we're doing this kind of development for, we sort of have this R&D validation going on with the clinical build happening at the same time. And with Mpox, where we had a pretty good design already available, some reagents we could get ordered, it took us about 4 weeks to get that up and running, which I think is, again, very impressive when you look at all the moving parts and pieces that are involved with this.
Final question. So back to the movie reference that we're in the middle of summer blockbuster season. I don't know if you have any aspirations of being a screenwriter later in your life, but if you were to do that, is there a particular category of microorganism or a type of infectious agent that you think would make a particularly strong story?
Yeah, I think actually the perfect pathogen movie has already been made. It was the 2011 film by Steven Soderbergh, Contagion. And he based that on Nipah virus, which is zoonotic virus, so spread from bats to pigs to humans, that's transmitted respiratory, so lots of folks get infected, and they can often present with these neurological sequelae. So it's a very striking clinical picture that really stays with someone. But the message of that film that I think really resonated with me, and is also just watching it after going through COVID, you'll see how prescient this was with some things. I mean, they mentioned social distancing in that film, is that the impact of these viruses, yes, there's a very human toll to it, but there's also this large social impact. And that's really, I think, where the true pathogenesis of the virus comes from, whether it's the misinformation that gets spread, sort of the distrust in your neighbor, the jockeying for who gets the vaccines first, all these very human conflicts that arise out of the virus itself. And so I think the way that he represented that, the way that he kind of represented the struggles of public health and the scientists was just incredibly prescient and much better than I could ever do. So if anyone hasn't seen that yet, go out and watch it and just be amazed at the filmmaker's skill in that.
Ben Bradley, thanks for being on LabMind today.
Thanks so much, Brian.
LabMind is sponsored by ARUP Laboratories, a not-for-profit enterprise of the University of Utah and its Department of Pathology. Our producer is Cherie Peterson, with audio engineering by Isaac Acosta Alvarez. You can also find other LabMind episodes at arup.utah.edu, along with an extensive video lecture library providing free CME and CE credits for medical and laboratory professionals. Subscribe to LabMind on Apple Podcasts, Spotify, or your other favorite podcast app, and please leave a rating and review in order to help others find the podcast.